50% of the arable land the expected or unfortunately expected that will be under drought affection. So currently, and also I wanted to include that 20% of the arable area is affected by soil. You know that this is our challenges. And despite all these challenges, another important part of plant breeding part of is biodiversity. Biodiversity is getting threatening every day, and then getting less and less. So creating genetic variability to contribute biodiversity getting much more importance for us as plant breeder, also world agricultural scientists. So despite all this negative effect, there is a solution. Solution is us. You know, you saw this week many young scientists here in agricultural scientists. That is our hope, and we need to help them. We need to train them with the to how to change these agricultural challenges, and then how to contribute food security, food and nutritional security in worldwide. So I will continue with the climate smart agricultural plant breeding methodologies which can be applied. It could be a good tool in our plant breeding programs to solve this problem from our plant breeding part of the project. So induced genetic variation is one of the very important tool as International Atomic Energy Agency. We are promoting this technology to create new genetic variability which is not existing in germplasm. The main advantage of nuclear technique, comparative advantage of nuclear technique is it can create new novel genetic variability. If you have already your traits, your genes, you don't need mutation breeding, you just cross it. But unfortunately, you saw a few slides before, we saw the challenges which we need to solve. So there is induced mutation breeding is one of the method could be applied in to solve this problem. So we are at the age of biotechnology. So without biotechnology, genomic tools and precise high throughput phenotyping screening technologies, molecular technologies, and also we are at the age of IA, you know, day by day, using AI technologies for predictive breeding. We need to increase more and more. This is the solution. This is the tools that we need to teach our young generations, junior scientists that can we will do. But I do expect that young generation will do better than us. That's why we need to train them, and we need to provide capacity building for them. So using AI data and to predictive breeding and using all genomic technologies is the solution. And the main solution is our young people and their willingness, and also us that we should train them, we should give them very good opportunities to contribute sustainable development goals of the United Nations. This is the newer genetic sources. You know, as currently we have more than 3,000 varieties, and most of them created by using gamma rays. But we have newer genetic, newer radiation sources, such as ion beam, such as proton beam, electron beam. They are much more precise sources to create genetic variability. Also, using space breeding is another technology which are being used longly by China. Also, at the joint FAO-IE Center, we are exploring opportunities how we can use space breeding to create genetic variability. That is another important source for us to solve this climate change problem. So two main idea is climate resilient, climate smart crop breeding varieties to develop for biotic, abiotic stress resistance with the good traits and good yield and nutritional quality. If you allow me, I will say a few words for comparative advantage of induced mutation breeding. And as I highlighted, first one is for creating genetic variability. Second one is faster. And when you compare to traditional breeding methods to obtain homozygous lines or stable lines, mutation breeding is much more faster. Third one is can be applied all crops, such as, for example, banana, vegetatively propagated crops, banana or potato, cassava, or seed propagated crops, we can apply for all. And the compared to comparative advantage of in that point of induced mutation breeding technique is, you know that, for example, banana has chromosomal barrier and almost not possible to use crossbreeding, and it produces very rarely seed, and that is very important that mutation breeding can create genetic variability using vegetative part of the plant. And that is another advantage. And also for some crops has different floret structures. It is a finger millet, subflower, which this kind of crops not allow to crossbreeding. Mutation breeding offers very unique opportunity for them as well. Fourth one is, actually there is many advantages, but I want to just highlight this one as well. Since yesterday, we saw many quinoa research that it is really hopeful to see that quinoa is very important crop and previously mostly cultivated in Latin America, South America, but because of the nutritional quality, it gained very good popularity. And many countries wants to develop these varieties, wants to bring their country. As plant breeder, and some of you I know that doing plant breeding, when you brought new material from outside, the most important problem is adaptability. What you do is you can do crossbreeding and to overcome this adaptability problem. However, in case of quinoa, for example, for my country or one of the country brought from Latin America this quinoa, how you can do crossbreeding? There is no wild species around. There is no local variety. So induced mutation breeding could be another option for this as well. Now I have to move. I know, time limitation. This is the summarizing the plant breeding activities. You know, induced mutation breeding is only different than the creating genetic variability from crossbreeding. As I said, homozygous lines is much easier. For traditional breeding, you have to go F2, F3, F4. But in mutation breeding, in 2 generations, you can get better homozygous lines. This is a big advantage. And combining with the biotechnologies, which I summarized in my previous talk, that using all these biotechnologies to develop new crop varieties this is the summarizing the whole step of the plant breeding to develop climate smart varieties for the future. And apart from that, just this is soil related, I'm plant breeder, and this is from my colleagues. What they do is they are using for nuclear technologies also to data driven. You know that if you don't know the data, so you cannot produce anything. Data collecting is very important. So using isotope and nuclear techniques to improve soil fertility, efficient water management, which is plant breeding is a tool. It's developing a variety, but we need good environment. If you think that new variety is a baby needs food, and soil is providing food. Also, we are using nuclear techniques, soil water management related as well. I just want to let you know the nuclear techniques importance. So this is the according to our database. This is a official IAEA database, but volunteer-based database. More than 3,400 varieties are being released and cultivated by small-scale farmers. As I said, this is volunteer, and for example, China, according to our records, has 800 variety, but I know that China has almost 2,000 variety, 300 from space breeding, which is still in process in this database. You might see that Asia is really being extensively used as technology, and our recent project in Africa is we are trying to increase the mutant variety numbers, but I checked that it is only or mutant or in Africa. Releasing new variety numbers is in traditional breeding is very less as well. It is not only for mutation breeding part. Despite this, we are working with many member states. I will show you many success stories from Africa now that what we are doing. This is from Vietnam. I selected this example especially for salinity purposes. Salinity is big problem when you cultivate. It's of course cotton or rice is especially irrigated crops, not rain-fed crops. Areas are much more affected compared to rain-fed crops. So in Vietnam, rice is very important. It is not only food; they also income bringing crops. And they have Mekong Delta, very big region, and cultivate the rice. But because of the salinity problem, and it is plant breeders work on salinity problem. Recently, they developed a variety, VND20. Yes, VN 9520 is high yielding, salinity tolerant variety. The breeder institution also awarded because it is increased their income. That is the reason. And Vietnam is a kind of icon for mutation breeding. Actually, they have more than 50 varieties, and these are most of them is cultivated by farmers. And in terms of rice, 3.5 million hectares in Vietnam, mutant varieties are being cultivated. 1 million is only this salinity tolerance variety. And this is from Thailand. It is about to release a variety. We developed with the Ministry of Agriculture, Thailand, this variety. As you see, it is submergent tolerance. We use electron beam, and almost the varieties are being released. It is in the final phase, and testing by for the registration and release of the submergent tolerant mutant variety, which is previously it was very important variety. We irradiated this sensitive variety with the electron beam, and now they develop or we developed new rice varieties with submergent tolerant. And another example is from Bangladesh. Bangladesh as well, they released 98 mutant varieties, and more than 20 rice varieties are being cultivated now. And they have one season, a manga season, and that is well known for unemployment. No crop can cultivate, so very bad season for them. They released a variety, it is 2012, I think. Since then, they cultivate rice variety, one rice variety there, namely Vinadian 7, stress-tolerant variety. Another one is Vinadian 20, is with the well-known with the nutritional quality, and also high-yielding variety is being cultivated by Bangladesh. Bangladesh has also released for chickpea, cowpea, many crop varieties. 30% of the chickpea area is covered by mutant varieties. And for mung bean is 15% of whole Bangladesh Mangpin areas, 15% is mutant varieties. So you may have heard that Pakistan has many good success stories as well. Now, currently, 30% of the cotton cultivated area is in Pakistan is mutant variety, which previously more than 50, but now it's 30%. And this is from Africa. I want to show you one more from Africa. That no, so far I did not show Africa. I want to show some from Africa. And we work with the Sudan and many crops, but especially we recently released a groundnut taproot variety, and it is 11% higher yield than compared to varieties. These ladies selected this variety. Actually, I went there. This is Elobeit region in Sudan. And the lady which is showing the groundnut is, she said, I selected, this is the best one. Even she started to some multiplied seed. So we discussed with the FAO. You know that we are joint FAO IEA center. So I also time to time we are working directly with FAO. FAO Sudan has helped us, and now they increased the seed. And because this variety seed is very good yielded and also good adapted, maximum 250 millimeter rain they have, and the yield was very high compared to the local varieties. I couldn't remember exactly now. So that's why FAO pay attention on that, and they help them to seed multiplication, because this lady said, if you don't give, even I will do. It was so good variety. So another example from Namibia. Namibia, we are working with Namibia, I think, since 2010. And they, prior to that, this mutation breeding program, they never had plant breeding program. Even now in the process to release variety, because officially it is still not, they don't have necessary law and regulations. And we work with this young and very hard worker people. I was lucky to having them. And we released in almost 10 years, we released 5 copy and 4 sorghum varieties, all of them at least 20-30% higher yield than local varieties. I know that as a plant breeder, 20-30% is very high to say. But even some areas 40-50, because why? They did not have any good variety. I was shocked, you know, as a plant breeder, and normally our aim is the maximum you can go 5% higher. When you say 20% higher, 30% higher, normally I don't listen. I said, oh, it is not, it's a story. It is not real maybe, but this is not a story. Since their variety is not long time, they did not have good varieties, and that's why compared to local varieties, at least 30% higher yield under drought condition, rain-fed condition, this sorghum and cowpea varieties. Now I go to Latin America. This is from Cuba. And Cuba also using a very extensively mutant varieties, and they have they previously cultivated more than 20 years rice variety. Now it's reduced now. Now it started tomato variety. Also from Argentina, I want to give you another very good example from Argentina. And they developed herbicide resistant variety long time ago, but the variety is very important, and even they use for crossbreeding. However, still more than 40% of the area is covered with this herbicide resistance varieties, mutant varieties. You may know that mutation breeding is very good for herbicide resistance as well. So, yeah, I think this is the last slide, if I'm not wrong. So, another example is from Peru. And Peru is well known for nutritional crops such as quinoa, such as amaranth, and they release the amaranth variety, and also they release barley variety as well for harsh conditions. And these varieties are being cultivated by farmers. My knowledge, 60–70% of the amaranth is in Peru is mutant varieties. And also I put this another small part of the banana related work, which is not directly addressing this conference, but I just want to let you know that we have many projects. Some of them is technology development. So far, use of capacity development project I explained you. Also, we are working for technology development as well. And current focus is with the banana, one of our project. And that's why I showed this picture, this slide that, you know, we have developed some lines with Fusarium wilt resistant 1 and 4 in Africa, also in Latin America as well. In the process now we are checking second time, and after when we get the confirmation, because you know that disease is sometimes depend on the rain and depend on the conditions. And even the lab conditions showed very great result, but we wanted to test at the with the hotspot, and then we'll let you know soon. So I think this is the last one for me. Oh, this is the future. This is for impact-based slides. It was last one. Now I want to talk with you. I want to discuss with you the future. So as I mentioned, that future is for me is first of all young. You know, many young people are here. That for me is the most important thing is to have young people that willing to work, willing to contribute, willing to take over this role what we are doing now. So first thing is climate smart breeding. Is we need to continue with climate smart breeding with the novel technologies, and is using AI tools, genomic tools, and bioinformatics and predictive breeding using AI tool is, I think, combined with genomics will be the in 5 years is the best solution for this climate smart breeding. Hopefully, even we can find find drought resistance despite this complex genome for salinity and also drought as well, hopefully, with this new tools. And increase mutation breeding efficiency, embrace novel technologies, and the most important thing is robust partnership. We are all here. You know, one African proverb says that if you want to go faster, go alone. If you want to go far, go all together. We have to go all together, and we have to work all together. So far, I think the main lack of the plant breeding project, all of the project is most of them not using holistic approach. We need to use holistic approach. All is in one. I'm working one part, in another one working another part, so that we can find a solution. You know, I have here my counterpart, Mrs. Habiba. You know, we are working together for Kuwait, and also Kuwait is working for other member states as well. So if we were go like that, you know, all together with the holistic approach, hands in hand, we can solve. I know we have very big global challenges, but solution is innovation, science, technology, and innovation, and invest the young people. Thank you so much.
Thank you, Dr. Fatma, for excellent presentation. And let us move to the second presentation. I think I will leave the question answer session at the end, because we have three. Okay. So we have three more presentations. So let me call Zainab Dagdad to present understanding the interaction of above and below ground traits in faba beans under different water regimes, a field study. You are given 12 minutes, okay? I will give you 2 minutes, okay, after 10 minutes, okay? So when I come here, it means 2 minutes.
Just to know. Good morning, everyone. My name is Zainab Dagdad. I'm a second-year PhD student at ICARDA with the University Mohammed V. I'm happy to present a part of my research focusing on understanding the interaction of above and below ground trait in faba bean under different water regimes, a field study. So I'm working under the Rootures, is a European project funded by Union European, focusing on root phenotyping and genetic improvement for rotational crops resilient to environmental change. As we know, thank you. As we know, agricultural systems are facing more and more constraints due to climate change. So addressing root trait is a real challenge for breeders that need efficient tools. In Rootures, we are developing a root phenotyping toolbox for field and controlled conditions to define some idiotypes for a changing environment and also to test innovative genotypes able to enhance the tolerance to abiotic stresses. To achieve those objectives, Rootures has an interdisciplinary team across Europe and Africa. In Africa, we have Morocco and South Africa. This distribution allowed the Rootures to study a large diversity of agroclimatic zones. At ICARDA, we are working mainly on Work Package 2 and Work Package 4. Work Package 2 serve to develop the phenotyping toolbox and to find some proxy traits for roots that can be useful to apply GWAS in a large diversity panel in Work Package 4. Here in the right, we have an illustration, resume the most important root traits that are under investigation by the project. So faba bean is a valuable but also a drought-sensitive crop, making the improvement of roots a solution to develop resilient cultivars. So to facilitate the study of this complex root systems in the field, the Chevlomex method was validated by the project due to for his low cost and also because this method they give us access to the top 20 centimeters of the soil, and we can assess the root architecture within this level. Here we have, in faba bean, we have an epogeal germination, means that the cotyledons are below the soil. This type of germination allows us to differentiate between epicotyl roots are above the cotyledon and primary roots and lateral roots below the cotyledon. This type of germination is the main trait that Chevlomex protocol is based on. So our objectives are to identify. We need to identify some above ground trait proxies to avoid doing Chevlomex in the future, because it's not an easy method. It's time consuming and it's a destructive method. Also, we need to select some main traits to apply GWAS in a large diversity panel of genotypes. Our experiment was conducted at field station of ICARDA Marchouch, located at Khmissat province, characterized by a semi-arid climate and also with a vertisoil type of soil. Our experimental design, so here we have 12 genotypes that it's a many core varieties selected from a large diversity panel based on their seed size. We have here major type and minor type. Those entries was planted in an alpha lattice design, and 2 treatment was applied, irrigated and rain-fed. Our root sampling was done at the beginning of flowering to be sure that we are digging and we extracted all the root system. So digging, extracting and cleaning are the most important step in this protocol, because we need to be careful with the root to avoid losing them. Especially in digging and washing. Washing, we need to try to be gently with the roots to avoid losing as much as possible. So then, since we have our roots clean, we can start the nodulation assessment. We have here nodulation number is an estimation of active nodule number. Also, nodule size, we can use those dots in the scoreboard are from 1 to 5 millimeter. Also, we recorded the number of active nodules. Are the pink color inside means that the nodules are still fixing nitrogen. Here we have the B nodules scoring chart that we are using to record the nodulation score. And here in this illustration, I tried to assemblate all the data recorded related to roots. We have root numbers and length and primary root branching. Also, we have stem width and taproot diameter. And at the end, we are taking also the fresh weight and the dry weight of root and shoot. Here, this slide shows how we can record the angle using the Chevlomic scoreboard. So we need, if we want to take the angle of our faba beans, we need to put the origin of root at the bottom, and we try to record the angle on the base of outermost roots in left and right. And here we have the horizontal and vertical measurement that we take, and also we are taking the root width max at the root width max at 118, and also we need to turn our root at 19 degrees to assess the root width in both directions. So here we have our agronomic data that we take during the life cycle of our faba beans. We have days to flowering, days to podding, days to maturity, plant height, and all data related to yield component. So here we have the broad sense irritabilities of our field trails. We have here the broad sense of our traits from the first year data, and for the second year we have only Chevlomics data because the plants are still in the field. We didn't finish the harvest yet, so we have only Chevlomics data. For agronomic data of our first trail, all the traits show high irritability up to 0.7. That means that we have a strong genetic control of our traits. And for Chevlomics data from first year trail, from the first year and second year data of Chevlomics data, we have the majority of the traits still consistent in terms of irritability, especially stem width and basal root number, maximum root depth, and also dry weight root. So here we have a big matrix. It's a percent correlation coefficient combined between treatment, show the correlation between above ground and below ground data. So here I tried to highlight the most important correlation. Okay, so here I try to highlight the most important correlations. We have stem width and plant height have the moderate to strong correlation with yield component and some root trait. Also, we have biological yield and green yield have a strong positive correlation with dry weight and fresh weight of roots. Also, we have here in the green square dry weight and fresh weight of shoot. They have a good, strong, positive correlation with dry and fresh weight of root. Here in this correlation is a correlation between Chevronomics data of this year. Some correlation are still consistent between years, especially stem with correlate strongly with with the root and dry weight of root, also with top root diameter, and we have a strong positive correlation between stem width and primary root length. So yeah. Then here we have multivariate analysis that show a distribution of genotypes on the base of the variance contribution of the trait. We have 3 clusters. Here we have the cluster 1 is an outlier, a genotype that is far away from the bottom of the PCA. And we have 2 clusters that are overlapping. That means they are controlled by the variance, the strong variance contribution of the trait, especially green yield, primary root length, stem width. So it's confirmed the result from our correlation matrix that those traits are the most important traits that we need to consider. So as a conclusion, here we have the useful proxies that we select and the selected root trait that we can apply in a GWAS study are stem width, basal root width, basal root angle, maximum root length retrieved, and dry weight of shoot and root to apply GWAS in a large diversity panel of 300 trees. Thank you.
Thank you, Zainab, for excellent presentation and also respecting the time. Let's move to the third presentation by Muhammad Arid. Can you please come forward? Thank you. So, we have 12 minutes, okay? 12 minutes. So I'll give you later on we'll do.
Okay.
Question later on.
Okay.
10 minutes. When I come here, 2 minutes.
Okay, thank you. Thank you very much for your attending this presentation. Hello, everyone. It's a pleasure to be here. I would like to sincerely thank the organizers for this opportunity and for the amazing organization. My name is Mohamed Zarid, Associate Professor at Faculty of Science, Ibn Tufayl University. Today I will try to present some of our results in this brief presentation entitled Differential Expression of Cucumis Milo TCP15 and GDSL in non-climacteric melon during ripening. As you know, melon is an important crop with diverse quality traits like sweetness, texture, especially aroma, and non-climacteric varieties such as Piel de Sapo ripen gradually and don't rely on ethylene. This makes it challenging to optimize both shelf life and flavor. So to address this, breeders use near-isogenic lines which allow precise study of individual genetic regions. The main objective of this study was to understand how these two genes contribute to fruit quality traits in melon. More especially, we compare their expression in the near-isogenic line AC102 versus PL de Sapo parent. We also assess how TCP15 affect ripening and firmness, and how the GDSL influence aroma compounds production. The melon near-isogenic line AC102 was obtained through interbreeding led by molecular markers between a Korean variety Songguan Charmy and the Spanish cultivar Piel de Sapo. And this near-isogenic line carries an introgression in linkage group 10 from SC into the Piel de Sapo genome. And the melon cultivation was under Mediterranean conditions in Torre Pacheco in Murcia, Spain. The flesh samples were obtained according to the Santos 2007 and stored in minus 80 degrees before freeze drying. The extraction was performed 2 times using TRI reagent RNA isolation protocol, and high pure total RNA was quantified with a nanodrop spectrophotometer with RNA quality verification by calculating 2 absorbance ratio and by electrophoresis analysis. To explore gene expression, we used RNA sequencing on fruit samples from AC102 and Piel de Sapo at different ripening stages. We also analyzed aroma compounds using gas chromatography, mass spectrophotometry, and these methods allowed us to link the expression of both genes with changes in ripening behavior, firmness, and the aroma profile. Our results show that both genes are more highly expressed in the near-isogenic line AC102 compared with Piel de Sapo, and TC15 expression increased especially at the late ripening stages, and this was linked to delay ripening and better firmness. Meanwhile, DGSL peaked in mid-ripening and is associated with a strong or stronger aroma, likely through its role in producing volatile esters. And so these genes appear to work together, TCP15 enhancing shelf life and GDSL enriching aroma, making the near-isogenic line AC102 a promising line for melon improvement. After identifying the roles, functions, we found that TCP15 delayed ripening through its role in jasminate, signaling, and cell cycle control, helping maintaining fruit firmness and extend shelf life. On the other hand, GDSL enhanced aroma by producing important compounds for flavor. Together, these genes show a complementary effect, improving volatile esters, both shelf life and aroma. This supports the use of targeted integration as a valuable strategy in melon breeding, especially in non-climacteric varieties. The integration of AC102 in the linkage group 10 was associated with 2 differentially expressed genes that can be associated with noticeable differences in fruit quality and delay ripening. So to conclude, our study showed that both genes play important roles in improving fruit quality traits in non-climacteric melon. TCP15 helps delay ripening and maintain firmness, while GDSL boosts aroma through ester production, and these results, as I said, highlight the potential of using targeted genes integration in breeding programs. Moving forward, evaluating how these genes respond to environmental factors will be key to achieve consistent improvement across different growing conditions. So this no. Sorry. For further information, this paper includes some of our previously published findings. We are currently working on integrating AI into our approach with the aim of obtaining complementary analysis and, if possible, optimizing their exploitation. And that's all. Thank you very much for your attention. Just, I would like to, I think I still have a minute, please, okay, for a question. As I told you, we are trying to use some AI tools. If possible, please, of course, I really need the feedback from such scientific community. I was discussing with a colleague, Professor Hmouni, about AI. As I mentioned before, we are trying to use some AI tools into our approach for complementary analysis, etc. So knowing that AI algorithms are based on data sets that remain limited compared to the vast diversity and probabilistic complexity found in nature and in biology, how much we can trust AI-generated insights in plant science and in science more broadly, knowing that experimentation has always been the foundation of the scientific discovery. Thank you very much.
Thank you, Mohammad Zarid. And the last presentation in this session is by Silmani Khail, and the topic is chromosome level genome assembly and functional annotation of Argania spinosa. Thank you.
Good morning, everyone. I'm honored today to be here at the third third edition of LAFOBA forum. So I am Sliman Khayi, a research scientist from the National Institute of Agriculture Research in Rabat. And my talk will walk you through the development of the first telomere-to-telomere reference genome assembly of the Moroccan Argan. Before to start, I would like to thank the forum organizers for this opportunity. So, to understand how this kind of project matters, we need first to start with the context of Argania spinosa. When we hear the word Argania, so we go directly to think about the argan oil, that is the precious liquid extracted from the argan tree nuts. And as a scientist, also the word Argania brings us to think about the resilience of the tree itself in this harsh environment of semi-arid and arid regions in Morocco. And when we say harsh environment, we mean not only low precipitation and high temperature, but also, as you can see on the image here, goats that are climbing the tree represent another kind of harsh environment for this tree. So geographically, the Argan tree is largely distributed in the west center of Morocco and not the south of Morocco, as largely reported in the literature. And as you can see on the map here, the green area is the distribution of Argan tree, and it is a UNESCO designated Argan biosphere. So from a biological perspective, the uniqueness of this Argan, of this plant species is that the only member of Phosboucraa family in the North Africa. So the Argan significance stands far beyond its biological uniqueness. It's play a crucial role in economic role, social role, and cultural role. Ecologically, this plant species contribute to combat desertification, soil erosion, and also the Argan forest constitutes an habitat for a variety of plant species and animal species. Economically, this plant provides food for human, livestock, and it's also the source of valuable argan oil, and it supplies woods for various uses. So Argan tree stands in the intersection between biodiversity conservation and sustainable development. Unfortunately, Argania spinosa forest area is in decline since the 19th century. So due to combination of several factors, climate change, overgrazing, overexploitation, and natural limited regeneration. As a response, and in part of the governmental Generation Green 2030 initiative launched by the Moroccan government, several goals has been set for improving and restoration of this argan forest. Among those goals is doubling argan oil production by 2030, planting around 50,000 hectares of new argan fields, what we call arboriculture, and also rehabilitate around 400,000 hectares of existing forest. So the main challenges for Argania spinosa are genetic variability. That means if we want to grow trees from seeds, of course we will have a high genetic heterogeneity, which implies that we cannot, which is impossible to maintain the elite traits, such as argan oil content, etc., and drought tolerance genes. Also, in addition to that, long juvenile phase, low regeneration rates. So the bottleneck today for arboriculture is the lack of high output approach to propagate the elite varieties that have been selected and registered. As an information, INRA recently selected and registered 8 elite varieties of Argania spinosa to support domestication of Argania spinosa in modern farms. So this technique, if we resolve the issue of the lack of this technique, we can ensure 2 important things: uniformity, that will enable mass propagation of elite varieties, and speed, that will help in a large-scale reforestation and orchard establishment. So, in the context of this alarming situation regarding Argania spinosa forests, we believe, as a scientist, that developing high-quality reference genome sequence for this species is the first crucial step if we want to tackle the several biological and scientific questions about this species. So here is briefly the workflow, bioinformatic workflow adopted by our institute to develop this sequence. So the workflow contains 4 main parts: genome survey, context assembly, chromosome assembly, and then we go forward for transposable element identification and genome production, gene production and annotation. So this slide shows the result generated from sequencing of this species. We combined the long-read sequencing, PacBio Hi-Fi, and short-read Illumina sequencing, and chromatin conformation capture coupled with short-read sequencing in what we call high-C data. So we believe that this data are a robust foundation for assembling this genome. So we previewed the first step in the assembly process of the genome sequence is to estimate genome size of the species. 2 approaches was used. The first approach is flow cytometry. The flow cytometry resulted in around 805 million base pair as genome size of the plant species. But when we estimate the genome based on camera frequency, as you can see in what we call here the genome scope profile, we have around 2 peaks with camera frequencies, which is a signature of a diploid genome, but the genome size estimated is around 645 million base pair. And with high rate of heterogeneity with around 1.48%. So this discrepancies between flow cytometry and in silico estimation of genome size could be linked to the fact that recently some colleagues from our institute described that for this species we have several variation in chromosome number for this species. We could have 2n equal at 20, 2n equal at 22, and 2n equal at 24. And that could be linked to this variation. Adding in as information, the individual used for flow cytometry is not the same that has been sequenced. We already published the draft genome, really a draft genome, highly fragmented genome in 2018 with low parameters and the chloroplast genome. But today we believe that we improved significantly this genome sequence. As you can see at the end of the assembling process, we have two haplotypes with around 17 contigs and 77 contigs, with high N50 matrix, around 50 million base in 50 and L90 around 10, and all these parameters are used to evaluate the assembly, and all these parameters here are of very, very high quality, indicating that we have a robust assembly of this genome. So this plot just try to summarize the statistics. So here we have high-C contact map. This contact map provide spatial organization of the genome sequence of Argan spinosa. As you can see here, we have each point on the plot indicates the intensity of the contacts between two genomic loci. And as you can see, the red, the blue square here indicates what we call TADs, topologically associating domain, which are the foundation unit of the chromosome. So the diagonal line here indicates 11 chromosome for the haplotype 1 and 12 chromosome for the haplotype 2, with a small chromosome here at the end showing some high frequency contact map with the chromosome number 2, and it could be linked to genome duplication. So when we talk about T2T phased assembly, we need to assemble the telomeric repeat in the chromosomes. As you can see in the plots here, we have, at the end, we have high frequency of telomeric repeats for each chromosome. So we thus confirming that we have T to T reference genome, except for chromosome number 10, we have only one end, and chromosome number 5, we have only this end. And the right plot indicate the synteny between, genomic synteny between Argania spinosa genome that we assembled and SHE3, the closest sequenced species to the Argania spinosa. SHE3, Vitellaria paradoxa, is among Sapotaceae family. As you can see, the outer ring indicate the 11 chromosomes, and the links between these two chromosomes indicate collinearity, synteny, and conserved gene order between the two species, adding another evidence for our high-quality assembly. For the annotation, we predicted the genes within this genome, and as you can see, we have around 60% of genome composed of high repetitive sequence. And we run a gene prediction. We have around 40,000 gene model predicted. As a preliminary result, within this 40,000 gene, we have 207 gene family now for lipid biosynthesis pathway. So I will go fast, don't talk about the phylogenomics. So this genome is not the end. Good genome sequence for a given species is not the end. It's the beginning of new era of organ research, conservation, innovation. And this resource, what we call genomic toolbox, will provide reference points for studying genetic diversity of different Argania spinosa population, because we need to assess the genetic diversity first and to decide what to conserve and what to restore. So genetic variation across population are essential for devising conservation strategies that protect full spectrum of Argania spinosa makeup, ensure long-term survival. Also, this tool facilitates genomic studies such as transcriptomics, proteomics, and epigenetics, where the studies provide deeper understanding of how species interact with environment and respond to stress and can be sustainably managed. Sorry for the time.
Thank you, Suleiman. Excellent presentation on Argan. Let's take about 5 minutes from the coffee break. If there is any question, please, can you raise the hand, and then we can ask the question. And the question is to whom? Which presenter? Dr. Fatma, question for you, please.
Yeah, thank you for the presentation. My question is that...
yeah, you can come here.
Not nice for me to...
Yeah,
sure.
Come here, please. No, you come here.
Yeah, you do, of course, mutation, or you create mutation just to create really variability. So I wonder, because you say 3,400 varieties released through mutation breeding, are these released direct release, or basically some of them were just this variability was used in crossing and all that, and ended in varieties? So because there could be some direct,
I see, I understood.
And then
some of them direct mutation, but most of them crossbreeding. Because mutation breeding, you can change one trait.
Yeah.
And then we need always crossbreeding. Most of them, it is combined as of, when you plant it, a few generations later, you notice the trait. And then sometimes this trait is not good for the, for example, you have resistance to disease, but yield is not good. Most probably we go either with the parent or the local variety, we do crossbreeding. So I can say that 70% is crossbreeding.
Good.
But when we have at least, parent, one of parent is mutant, we identify that this is mutant.
Thank you.
We accept.
Thank you so much.
Thank you.
The other question is to our colleague that from Ibn Tufail that on the melon.
Yes.
I wonder how useful, the finding are for Morocco, because I don't think we have a breeding program on melon in Morocco. I don't.
Muhammad Arid, are you here? Muhammad Arid, second presenter.
Because that could be useful for some.
I think he left for the coffee break, so maybe you can have discuss over there. Any other question, please, to
no,
the
that's
presenters?
thank you so
Yes.
much.
Your question is to whom? Fatima, thank you.
For the breeder. Okay, thanks so much.
Smart question, smart answer.
Yeah, quick question, yeah. I would like to ask, two things, just quick ones. The first one is how your organization is really giving service or collaboration related issues, like genotyping service, how you can give it like your organization to give like this making mutant for other researcher, one. Second one is what challenges, you might face during, these things, your collaboration, not yours, but other breeders, maybe when they are using your technology. Thanks. Very quick one.
I think first question could be very long. Maybe you can discuss during the coffee break. Okay, it's not for her, it's for the IAEA. And the second question, maybe you can answer, Dr. Thank you.
That is a challenge. I don't think that it is a no challenge that we cannot overcome. This is the short answer. I think you might hear this. So no big challenges actually when you send the material to us, or for example, in this region, Tunisia has sources. We can ask them to make irradiation for you. If there is a project with us, we cover all the cost of this irradiation services. But if you don't have project with us, it can be depend on but we can facilitate as a human, we know that we can facilitate. We have many different collaboration opportunities. It's good, we can discuss. As a doctor said, it is very broad.
Thank you. Is there any question to the first presenter? Do we have any question for the first presenter?
I have a
Zainab?
question for the second presenter.
Second presenter, okay.
Yeah.
Okay, so this will be the last question because we are running out of time. We have to go to the coffee break and come back 11:30. We don't have much time.
Yeah,
So
I'm not
the
sure
last
if
question, please. Any other question, please, you can discuss during the coffee break. Thank you. So what is the question now? Yes?
Yeah, thank you for the presentation. I was just wondering how useful your findings would be for Morocco, because I don't think we have a breeding program on melon, because those findings would be probably useful for, countries that they have programs on melon, breeding programs. I don't think we have one in Morocco.
Thank you very much for your interesting question. Simply, this is why I'm here in Morocco, because really, actually, I don't have a clear idea about what the colleagues are doing here in Morocco about breeding, but we have a diversity of varieties to working on, and this is why I'm here in Morocco. I decided to come back in Morocco. Thank you very much.
Thank you. After this, we can just conclude the session. Thank you very much for your presentation and your patience to listen these lectures. So let's go to the coffee break and come back 11:30. Thank you very much.
Moving to results, and as you can see here, we tested three machine learning algorithms. Starting from the left, we tested the GBR, it's gradient boost regression, moving to random forest, and finally QBIS, which is obviously having with this machine learning algorithms some reliable and robust results with R square of 86%. And in the bottom of my slide, you can see some features importance that are splitted into models that are come from 3D feature space. In yellow, you can see some spectral indices, and with green, it's principal component analysis coming from Landsat. In this slide, you can see our final maps coming from different machine learning algorithms. You can see in the right the final result coming from QBIS model with 86% of the error square metrics. And we have done some statistics to visualize the repartition of our salinity classes. We believe that integrating machine learning algorithms, remote sensing data, and soil sampling can give better and make us understand the salinity behavior in arid and semi-arid area. Second, we can use our results as a decision-making tool for our partner and stakeholders. And let me thank you for your attention, and I will be more than happy to answer to take any questions and suggestion or further collaboration moving towards. Thank you.
Thank you, Ayman. Now we have almost 13 minutes before we hand over the room to another session. So may I request all the presenters please to come here on the stage so that we can start the question answer session. Thank you. Dr. Ali So let us start from Dr. Ali Batai. So if you have any questions, please, to the first presenter, Dr. Ali Batai, you can please put the question. May I request you please put smart question and the smart answer so that we respect the time, so that we can hand over the room to another, another session. Thank you. Yes. Is there any question to Dr. Ali Batai for the first presentation?
Okay. I have a question.
You can ask the question, yes. Okay, go ahead.
For him, yeah. So you have explained, I saw your presentation, it was nice. Yeah, a lot of outputs, and also for all your team during these days. Just yeah, in your work you said you have used machine learning to enhance the output. It's just to explain to everyone how does it work with AI in this, because as old school as classics, like I learned how to do the mappings or using the satellite images. It's like it's CRAYGEN. We use CRAYGEN, and we use supervised classifications. In supervised classification, there are iterations and stuff to get rid of some mini layers and to classify. Okay, how does AI enhance this? Because when I saw the diagram, okay, it showed that it's like a kind of iteration. And can you explain more? Okay.
Yeah, Thank you so much for the question, Jamal. I think with the AI right now is not like it's not a luxury. Like this is like where the digital image processing is moving. Hyperspectral data has been around for at least 2 decades. The first product for hyperspectral, but they have not been very successful. Like used because it was very computational capabilities are very heavy at that time. So right now, with the emergence of these tools, machine learning is one of the basic things for the AI, and then deep learning, etc. So it is like in a way like what Ayman has just shared with us right now. It is the present, and it is the way to try to take the maximum of these features or this information within the hyperspectral information. We use the first derivative and second derivative both for reflectance and absorbance, trying to reduce the collinearity. Definitely, there is so many models that we have tried for the machine learning, to see which one is giving the best result. And this is what we have shared. So I think, going through the details on how to make this, or to explain it, it's a part of the digital image processing, course or class, which is not very relevant in this context. But as I said, with our team, we do believe in this next generation, trends of digital image processing. We are investing a lot in terms of human resources. All our students, they are getting these skills for the processing. And also, just to add and to share with the audience that in the Center for Remote Sensing Applications, we are planning, and we are very soon getting a hyperspectral drone, for the full spectral, visible and near infrared and shortwave infrared as well. And this will allow us, to understand more this hyperspectral data, to produce our own data, at high resolution, and to have a control on the noise and to have a control on the processing. So
thank you, Doctor.
Yeah, thank you so much.
Thank you, Doctor. Any other question to Dr. Ali Batai, please? If not, then we can move to Mr. Dinesh. Any question for Mr. Dinesh, the second presenter?
Yeah, I have question also. Sorry. Sorry,
Let us take from them, and then we'll come back to you later on. Any question, please? Okay. To the third presenter? Any question to the third presenter? Yes, please, over there.
Thank you. Please, thank you so much. So I'm very delighted to be with you in this forum. I want to thank all the presenters for their high quality presentations. I have 2 quick questions for Mr. Al-Halla. The first one is about the implementation of the model of the general soil salinity mapping. About the implementations, you have said that it is a function of several parameters like climate, other soil parameters. I want to ask you about the details about the implementation of the model to have the final soil salinity map, and the other presentations is how they proceed to validate the obtained maps in large scale salinity. Thank you so much.
Okay, so for the model, it's a basic model. It's the Gini model, we call it. It's known for anyone who studied soil science. He knows that soil genesis, there are 5 factors. Yeah, 5 factors. It's parental material, climate, topography, including slope and altitudes, and also organisms, and time. This is a big factor that we can't put into consideration. It's really very slow. So these are 5 factors. Here I showed more than 5 because I showed other factors linked to organisms like human activities. It's also few minutes. So these factors can be turned to maps. For example, for organisms, we can do a soil cover map. It's easy to do nowadays, yeah? For parental material, you just take your geological map, okay, and analyze it and turn it to lithology and see what are the most classes you can convert them to so that you will fit your output scale. You should put scale first, okay? And so on for the other factors. And using GIS, like QGIS or whatever, you can overlay them, okay? You have attributes and you have your units, yeah.
Thank you, Jamal. Thank you. Yes, please. Your question is to which presenter?
Ahmed Bouaziz. I would like to ask a general question about the link between mapping and agronomical practices in terms of salinity or in terms of erosion. What is the link? Because if there is no link with agronomical or technical management, what's the usefulness of the mapping? I give a practical example on the basening of the soil surface to increase the roughness of the soil in order to alleviate or reduce runoff and erosion, or use of organic matter in order to increase the soil stability. I don't know if my question has a sense for you. The link between mapping and agricultural practices. Thank you.
Thank you, Doctor. So your question is to which presenter?
For which presenter?
I can give an element.
Yeah. You want to give the answer, or should I give another lecture on this?
I will give a part of, for the part we are working on in Mutation. So just to give two examples. In the first day in the keynote speaker, we have heard the necessity of applying, let's say, water harvesting, techniques. And although those techniques are well established, they are still not adopted by farmers. But let's say in Mutation, if we want to apply this water harvesting, this is a very, as I explained in my presentation, is it a natural depression? So it is best suitable, and it is a semi-arid area. Best suitable area, to implement this water harvesting techniques. But if you have a problem with the salinity, then you have to make sure that the water you are harvesting is a good water, to be used for agriculture. And this is a direct link with farmers. The second, example, just to be quick, is in that area, people are getting groundwater right now up to between 300 and 500 meters. And previously they used to be 80 or 120 meters. Right now this water is saline. Then if they want to apply some water filtration or inverse osmosis or whatever techniques for desalination, then they will have a brine as an output. So managing this brine should take into effect the existing salinity of the area, not to aggravate this. This is only two examples. Maybe Jamal has to add something, or
I think salinity, we say if the salinity can be measured, it can be managed. So salinity assessment is essential in order to make some recommendation at the regional or country level or at the farm level. So there is integration approach. There is no single approach to handle the salinity. It could be from physical method, from chemical method, from the biological method, from hydrological method. So it is recommendations are site specific, location specific. The farmer may have one problem, he may have many problems. So based on the diagnostics, so if we do the proper diagnostics, and then we can make a better formulation for the farmers to adopt in order to improve the farm productivities. So we are running out of time now. Last question. If you have any last question to this last presenter. Yes, please.
actually is not a question, but just like complement of answer why soil mapping is critical, for example, for erosion. So one of the things that is like useful in remote sensing, we can generate digital surface model. We can generate we can derive lots of attributes about topography. So the slope, the areas where water can accumulate, all those things is very, very important for soil, and we can get them from remote sensing. And what Ali he said about AI, we can use the AI to predict many attributes for soil, like organic carbon, NPK. Those things we can have them through remote sensing studies and mapping, and from there we can make some recommendations, suggestion for the agricultural precision.
Yeah. The most important thing is that the message must pass to the farmers. So we have to really strengthen the link between the researchers, the extension workers, and the farmers. Any research done at the research level not going to pass to the farmer, there is no use for that. So we have to really strengthen the extension services to link with the farmer and the researchers, so that the technology developed must reach to the farmer immediately, so that the farmer can take the advantage, because farmer is actually the final stakeholder for all the information. Salinity is not a constant property, it's a dynamic. Today salinity may be very high, tomorrow there is a heavy rain, and salinity will be very low. So in this case, you have to live with the salinity. You cannot only measure salinity only one time and make a recommendation. No, you have to do salinity monitoring as well, in the plots or in the country level or at the region level. So salinity mapping, monitoring, and then modeling, and ultimately making decisions based on the scientific information is very important. And if the information is not going to the farmer, we are not doing our job. Thank you very much for your listening and patience. Now we hand over the session to the another session. Thank you very much, all of you.
So we're going to follow up with the next section. My name is Ahmad Lamrani. I'm professor at Center for Remote Sensing Application at UM6P, and I will moderate this session on best crop practices to cope with the abiotic stress. So we'll have one keynote presentation and 3 other presentations. So I want just to make sure that all the people they're gonna present, they are here. So is Miss Wisal with us? Okay. Mr. Sharqi Ghulam. Okay. And Miss Habiba Sood. Okay, we have everybody. So we're gonna try to respect the time. For the keynote presenter, we have Dr. Mina Divokota. She will talk about agronomic innovation for enhancing productive and resilience of dryland production systems. The floor is yours. Please make your presentation within 20-25 minutes. Go ahead.
I think I would prefer to have a mic. Good afternoon, everyone, and you are very welcome to this presentation. Today I will be presenting that agronomic innovations for enhancing productivity and resilience of dryland production system, mostly from research conducted by ICARDA. So most of the research I have been presenting is from our own experimentation. So, I'm not going to very detail about, what are the challenges for drylands. This is really well known that we have more than 40% area is under landlands and majority of the people are under. But there are several challenges, definitely climate change, water scarcity, loss of biodiversity, land degradation, the high population. These are the really challenges. And also pushing for scientists to grow more for such kind of environment. So that's why all these demands for the system, for the farming system to sustain the regions, food and nutrition security, and as well as for climate security too. So these are the really challenges for us. Just I would like to start with my presentation one. Yes, there are several technologies which has been already developed, and some are very, very interesting for the scientists, but scalability is very low. Why this is happening? This I always emphasize that, whatever we do, research should be in the core thinking of farmer and heart. Yeah. So this is one example. This is from Marsus. I think it was in 2019 somewhere. It was a dry year too. Our aim, agronomists, we want our crop like that. Yeah. But even in the same year, why some farmers has really good field and why some has really low. So this is our task to identify what is making difference. And in this context, I was analyzing one of the large data set that ICARDA has taken on 21 provinces of Morocco in major wheat growing area. And I had just figured it out that, okay, those farmers who has really don't apply any really farmer practice, it means like a very old varieties and very poor management thing, where the average productivity was less than 2 tons per hectare. This is all on rain-fed condition. But when I look different component analysis, I can see that, okay, if we even in the same year, if we have like a good management practices, we can get easily 1 ton additional yield. It means really good for this kind of environments. So why we agronomists has to focus that how we can bring from this less than 2 ton to at least 2.8 or 2.5 ton. This is our job, and this all begins with a simple thing that which we miss. Yeah. So most of the my presentation will be based on this line. And definitely it's a very limited time. I cannot present all this kind of thing. I want to list some of the agronomic innovations that we are trying. Conservation agriculture, crop diversification, efficient water management, efficient nutrient management, and combining genotype, environment, and management, which is really, really, really important. And when I see most of the breeders and agronomists somehow not always together. This is I have seen in most of the area, and which is happening always. So we have to be together. Not only the breeder and agronomists, but I also want socioeconomists, even the environmental. So all these things has to be together. Otherwise, we are not able to solve the problem. And definitely precision agriculture and digital agroadvisory, and also one I missed, the smart mechanization. I will say scale appropriate mechanization. Is also one of the agronomic practices that we are focusing in. So here I will just give few examples in this short time period. We all know that now conservation agriculture is also one of the very regenerative agriculture practices for resilient drylands, which has 3 major principles. Definitely the reduced tillage or minimum tillage, and residue retention and proper crop rotation are the major pillars for conservation agriculture. So we have several results because it has been started since early 90s. Even most of the countries are even already started adopting, and they have several benefits. For example, improving the productivity and stability, soil health, and saving the energy and labor use efficiency, water use efficiency, and profitability. So these are the, most of the benefits we can see from the conservation agriculture. Here, I would like to, ICARDA, together with the national and international partners, had significant role for introducing and developing science-based scalable solution for conservation agriculture, especially in Siwana region. Siwana means Central Asia, West Africa, West Asia, and North Africa region since early 90s. So this has several scientists has put a lot of effort together with the national and international partner on this line. And this is just one example I would like to give, that we have established different research hubs across the different country. And one of the example, definitely always we do together with our national partners, national research centers. One is in Morocco, in Marsus Research Station. We have more than 12 years of conservation agriculture and evaluating different innovations on that land. And similarly, Egypt, we have also started, because in the beginning, people were reluctant that, okay, in irrigated agriculture, we don't need conservation agriculture, it's okay, yeah, it doesn't work. But since 2-3 years, really, even Egypt, scientists are really convinced and now started, oh, really, madam, it works, even in irrigated area. So we have also started in Egypt and India in collaboration with the ICARDA. We have also, this station. And Uzbekistan also we have, and also Lebanon we have started, but unfortunately with this war, it's not moving that well. But we have this research hub. And also, we also generate all the evidences, what is happening in the region, how it is impacting on different indicators of conservation agriculture. So this is like a compilation of the study we have made for this MENA region. And then another one good thing is all these things putting together, helping us for the policy that's the now government of Morocco came up to reach 1 million hectare, and we have been supporting with the national partners, supporting to this ambitious goal through the different program. So here I just want to give very, very short, benefits of conservation agriculture. This is from North Africa, from 5 to 7 years of research under different country, and just I have summarized that how the conservation agriculture is helping for different indicators. Similarly, you can see this is an example from salt-affected irrigated drylands of Uzbekistan, and we have compared, the rotation system of cotton, wheat, and maize. This was on cereal and legume rotation system, and this is on cotton, wheat, and maize system. So you can see that there is all the benefits. So what my message here is, it is working both under rain-fed and as well as even irrigated and saline condition. So this is the main key message, and the paper has been already published, and you can go through it, and if you are really interested, I can provide the link. This is another example. It was in 2021 in Morocco. Again, we were evaluating in different crops, and you can see that, how the conservation and conventional system has helping to improve the productivity as well as the, the sustainability, stability. So this is first example is on lentil. I forgot to put exactly the area, but you can see the bag, and it is, lentil yield was almost around 63% higher yield, and similarly bread wheat, you can see that large bag and no till, and then small one is with the conventional tillage 66%, and for durum wheat it was 52% higher yield. It was, rainfall was on that year was just 250 millimeter in Marsus, so it's all under rain-fed condition. So why I'm saying all these things is, yes, if we do practice all principles of conservation agriculture, definitely we will get really, really good result. And I forgot to include one picture. This year was really interesting year for me. We planted, in November, somewhat 21, and there was few rainfall. And after we planted, there was just 7 millimeter rainfall. And only the plot where we had a conservation agriculture with wood was green across all this 150 hectare area in this region. So why I'm saying, what is this making difference? In my opinion, it is all because of we build up all this healthy environment. Yeah. So this is a really good thing that learning we should to be doing in the farmers field too. And this is a really one example I would like to show again, the experiment that I showed before from the salt-affected irrigated lands of Uzbekistan. And you can see there was actually, this conservation agriculture was under permanent raised bed, even not on the flat, because irrigated condition, you need to also improve the water use efficiency. Yeah? So experiment was for 3 seasons, and so it was, you can see the, and then salinity in, location is sulfur-affected area in Uzbekistan, cropping system is cotton, wheat and maize, and agriculture practices is permanent raised beds with and without residue, and then conventional tillage. And then another one is salinity level of that experimentation area is 8 to 10 decisiemens per meter. So what information I would like to give here is, yes, it works well, but it needs to be with residue. If you don't have residue on top, on beds, forget about it, because it evaporates. Still the, if you see, the rate of increment is even higher if you don't have residue on the bed. So this is main message is, yes, if we do the conventional practices, the salinity level will get increased. But if you use a conservation agriculture practices with residue retention and rotation, and it helps to decrease the increasing rate of salinity. So this is one of the good finding I have been suggested, and this is a top 10 and also the top 30 centimeter soil profile. Again, this has been already published, so if you are interested, I will be happy to share and some more detail. Yeah, I mean, just I would like to give a little bit here is, yes, opportunity to scaling conservation agriculture in Siwana region, yes, about 40%, 21 million hectare out of 53 million hectare arable land is suitable for CA. So we have done some kind of mapping, and definitely increasing young generation, they have interest. They want to adopt new thing, so there is still scope. And most of the agriculture operation is mechanized. I mean, I'm talking about, this MENA region. So it's possible. Most of the country has priority focus on climate adaptation and mitigation, and also the opportunity to link with improved land degradation. So there is scope. So just we need to do rightly and encourage the people. This is my, really suggestion for every agronomist. We should work with the farmer, convince with them, do rightly, and definitely we will reach one day, like Australia, 90% area under conservation agriculture. We can reach on that point also for the MENA region. And another thing is, I don't want to go very detailed here, diversifying the cropping system for sustainability in drylands. Definitely more than 80% area is under cereal monocropping. So if you see the picture here, area under legume is significantly declined in the recent years. But if you look the demand, it's not because of not demand, it's very high demand. Look how much is the import. So there is a really challenge that we should to think about. Why we are not only exporting, but also exporting the fertilizer, how much we are reducing. So this is a really big challenge. When I analyze this data, I feel like, oh my God, we should work on diversifying the system. Why is not going? Why it is declining? Yeah? This is the key message, and definitely I don't want to go very detailed on the advantage of diversifying the system. It's very clear. And how ICARDA is working is we have been evaluating different crops, crop rotation and crop combination with different agronomic practices, both on the conservation and conventional system. In different countries in collaboration with the national partner, and also it's to understand its performance, not only from the crop perspective, we have to look from the system perspective always. Yeah? So this is all objective, and we have been introducing several crops in the rotation system. My quick observation, my 7 years of experience is showing that at least we have to imbue 33% of either legume in the rotation system is more beneficial, because we have this rotation system experimentation. And just I would like to give a little bit highlight that, okay, this is again the same data that I have been analyzed that, and also we have asked that what was the previous crop on that particular wheat. You can see here the study was done both on the irrigated and rain-fed system in Morocco. So you can see the yield advantage, where this is the wheat yield. And we had asked, what was your previous crop on that particular field? So farmers who has adopted, legume in the previous crop has around 17% higher yield of both yield both on the irrigated and rain-fed system. So it is also farmers' condition. It's not only research station. And here I will just give one example. It was again in 2021, which was very, very, very dry in the beginning. So you can see that, how was the crop, wheat crop, where there was a lentil, there's a previous crop, and then cereal is a, in the continuous cereal, and you can see good impact, and also I have done also in the good year. So there is yield advantage. Yeah. Yeah, okay. And another one, I just want to give example that where I was mentioning that how we can intensify and diversify the cereal system. One is rotation, and another one is we were trying that, this is the way that how farmers plant lentil. They plant two, in Morocco, especially in Marsus region, they plant 2 rows together and leaving around 1 meter space, especially for managing the weed. Yeah, and in Morocco in rain-fed condition, we have a very narrow planting window, November till May, June. Only this window we have. And then we have introduced, I just realized that why not we use this space to plant another crop so that you can best utilize the available water, during that season. So we have introduced many different crops in between planting lentil. So lentil was planted somewhere in beginning of December, and second crop was planted end of February, because this, and then you can see the way how we have planted. I gave example how I planted quinoa in the standing lentil. So it was really, really good example, and this the year was very favorable. After there was a very good rain in the late season, so it's a really good option to harvest 2 crops. I just done some of the system level analytics for both. An example with lentil, wheat and lentil sol, lentil plus chickpea and lentil plus quinoa. It was really beneficial, and I found that definitely instead of having 1 crop, farmers can easily harvest 2 crops. Yeah, and then because utilizing the whole season rainfall, and also it minimizes the risk. If you have a really early season drought, if you lose the lentil, and if you have a late season, then definitely you'll have another crop too. So this is something I feel like a very effective risk minimizing option. And definitely if you have most of the time I have harvested 2, but at least 1 year, otherwise I have at least some. And high protein yield, definitely if you include legume in the system, and definitely diversify crops in the field, and also diversify the food in the table. Yeah, so this is really very, very important. And reduce application of chemical fertilizer, especially for the following wheat. So we have measured, this is all on the system basis. I always do on the system basis. So next year we plant wheat on that, and how is the impact in the following wheat. So we found that at least around 12 to 13% higher wheat yield when you practice this intercropping and compare to the cereal rotation. And also there is no yield penalty for lentil. Good thing is there is no yield penalty for lentil if either you plant intercrop or is a sol, because we are planting when lentil is almost grain filling stage, so there is no competition. This is the one thing I would like to highlight, and this innovation has been already identified as one of the sustainable land management practices, and then we have a lot of publications already. If you are interested, I will provide more detail. I have been testing this in 6 years, so a lot of information, many crops we have evaluated. Yeah, and again, since 2 years, I have been evaluating this innovation under conservation agriculture. These first two figures you can see, this is really interesting, and we have got really good result. Last year also we had a very good result, and this year it's also a very good result. These are the actually pictures from our shoes we have conducted. And also, beside this, we have been using to diversify low water requiring nutrient-dense crops, different crops, 16, 17 different crops, to see how it is performing, how it is fitting the system, which one is, is better. So this is the thing we have been evaluating. If someone is interested, I'm happy to provide all this detail. And if some students are interested to work on it, we are also happy to host. And also another one is definitely supplementary irrigation is to minimize risk. Always, if you see in Morocco, continuous 6 years of drought we face all the time. Always risks. If we look in the farmer field, hardly less than 1.5 ton wheat yield they have been using. So this year what I have done, these two pictures, I have a very good insight actually. There was 16 different crops were evaluated, and one I put just supplementary irrigation. This year was very dry in the beginning. Only adding 25 millimeter of water 2 times, 12 millimeter and 12 millimeter, 24 actually. You cannot imagine how is the field and how much will be economic benefits. So this is somehow giving us some criteria rather than failing each and every year. I think government is also planning for that. This is a really good result that how we can optimize in this kind of thing. And definitely I don't want to go there very detail. We have been working on different other water management practices. And one other thing because I realized that this is a salinity, and why not I present this kind of work. It's very short. Efficient water management to manage salt in salt-affected lands. This is again a study from Uzbekistan, and there was a 3 irrigation, and the location was, salinity level was more than 12 decisiemens per meter, and crop was cotton. Planting method was raised beds with 90 centimeter furrow to furrow. And we had implemented 3 irrigation methods. One is irrigate every furrow, and another one is alternate, ping pong. One time one irrigation, one time another. And another one is permanent scape. Just irrigate only one furrow and keeping the another furrow always dry. So there are 3 methods. And you can see that this is the principle, how salt ping pong across the different, irrigation methods. And, okay, I forgot to say in Uzbekistan, in the high saline area, before planting, they use, you cannot imagine, about 20 centimeter water depth just to leach out the salt. And then shallow groundwater level, and they are increasing the secondary salinization. It's a really big challenge. That's keeping in mind on this aspect, I have tried this experimentation besides my PhD thesis by that time, and this experiment I did too. And then definitely water they flood even, very harshly in both, in farmer field. So this is all is causing soil salinity, secondary soil salinization, and also raising the groundwater, and definitely water, this precious water, how they are, So, so I think these 3 methods is clear. Okay, and also, yeah, I'm almost done. Also, there were we did a leaching because I said to you, if you don't have a residue on the bed, salinity will increase. So in this case, we also use a leaching technique. After 3 to 4 irrigation cycle, we apply the leaching technique differently. Like for every furrow irrigation and alternate skip furrow irrigation, we applied water at the same time in each furrow. You can see, all furrow we applied water at the same time. But in permanent skip furrow irrigation, we started for wet furrow first, and then we applied water in the dry furrow, because most of the salt got accumulated in the dry furrow. I will show you. Sorry, yeah. So this way, it's difficult to explain in this very short time, but it is really, really interesting, good result out was coming. And then also I evaluated all the cotton performance. It was very high saline soil, as I said, more than 12 decisiemens. So cotton yield was improved. You can see that in permanent skip furrow irrigation, we have around 2 ton, but in other, you can see 1 and 1.2 ton, yeah. So this was the salt. You can imagine, these are the 3. This is a before leaching. After 2 to 3 irrigation cycle, this is the salt concentration, and, in a different bed, because I said it's 90 centimeter bed width. And how was the salt? So in every furrow irrigation, salt was concentrated on top of the beds. And if you do an alternate furrow irrigation, it's ping pong. But if you go on permanent skip furrow irrigation, most of the salts were accumulated on dry furrow. What happens after releasing? Oh. What happens after leaching? Yeah, we leach the salt. It minimizes, rather than applying 20 centimeter of water logging for 4-5 days, we applied very limited water. And you can see, okay, in every furrow irrigation, and still salt are ping pong on top of the soil. But in alternate furrow irrigation, still it remains in the center of the bed. But if you look on the permanent skip furrow irrigation, salt were accumulated in one side. Yeah. So this is something, a permanent skip furrow irrigation technique with proper leaching reduces the salt concentration on top and side of the beds. So only one more slide. This has been already published. If anyone is very interested, I am very happy to share and share. Yeah. So my most conclusion is yes, on irrigated drylands, on salt-affected irrigated drylands, definitely salinity on top of raise will increase when irrigation applied in both furrows. And salinity level on the irrigated side of the furrow was always low, so that we can plant the crop which are salt affected, even it is options for diversifying the crop. So a lot of opportunity has given, lot of questions has opened with this innovation. So and my take-home message is the permanent skip furrow irrigation practice could be possibly more beneficial to farmers if salt-sensitive crops planted on the side of the irrigated furrows and salt-tolerant, less water-requiring crops on the side of the dry furrows facilitate also to diversify the cropping system. And also, further research is, however, needed to identify the combination of salt-tolerant susceptible crops varieties to cultivate raised beds with the permanent skip furrow irrigation and its benefits to the farmers and also to the environment. Yeah, I think I'm done. Thank you. And this is my quinoa field. Those who are quinoa lovers.
Yeah, thank you very much, Dr. Mina, for this interesting presentation. Very interesting. So we're going to keep the questions after the last presentation. So now I invite Ms. Wisal Ben Afri to come and talk about her work on quinoa. The floor is yours.
Aimable assistance, bonjour. J'ai l'honneur de présenter un essentiel de mon travail s'intitulant « Rôle des champignons mycorhiziens arbusculaires et des PGPR dans la médiation de la tolérance au stress hydrique chez les graines de quinoa en modulant les composés phénoliques, les marqueurs de stress et l'accumulation d'osmolytes ». Comme vous savez tous, l'eau est la molécule essentielle pour tous les êtres vivants sur cette planète, y compris les plantes. Cependant, il est actuellement menacé par les conséquences des changements climatiques, provoquant ainsi une augmentation de température et diminution des précipitations. Ces variations extrêmes provoquent plusieurs pertes de terres arables d'environ 12 millions d'hectares de terres arables, ce qui limite fortement la croissance et la production de plusieurs cultures. Ces variations extrêmes affecteront sans doute la demande alimentaire qui doit être augmentée en parallèle avec une croissance démographique en forte croissance, ce qui augmente la demande alimentaire de 59% à 98% d'ici 2050. Donc, le climat méditerranéen en général et le climat marocain en particulier incite le gouvernement, souligne une grande menace de la sécheresse qui est devenue une véritable menace pour le pays en général. Selon l'Institut des ressources mondiales, le Maroc figure parmi les pays condamnés à affronter un stress hydrique extrême avec des pertes de rendement qui peuvent atteindre 50%. En effet, le stress hydrique affecte négativement les propriétés physico-chimiques et biologiques du sol, par la réduction de la disponibilité de l'eau, l'appauvrissement des sols et la réduction de la croissance et l'activité microbienne. De même, il affecte négativement les caractéristiques agrophysiologiques et biochimiques de la plante en réduisant son état hydrique et nutritionnel, en perturbant les processus morphologiques, physiologiques et biochimiques et tout en réduisant la productivité agricole. En effet, le stress hydrique incite les gouvernements du monde entier à envisager l'incorporation des plantes appropriées aux agroécosystèmes menacés tels que quinoa ou bien Chinopodium quinoa, qui sera un allié contre la famine dans les pays, dans les zones arides et semi-arides. Cette pseudo-céréale qui est peu reconnue, elle contient des grandes quantités en fibres, en vitamines et en antioxydants qui lui donnent son impact substantiel contre la défense de plusieurs maladies telles que les maladies neurologiques et les maladies cardiovasculaires. Outre l'adoption des cultures résistantes aux stress environnementaux, des approches durables, écologiques et innovantes doivent être mises en place pour augmenter la productivité et la croissance de cette culture, notamment l'utilisation des champignons mycorhiziens arbusculaires qui boostent la productivité et la croissance des plantes en améliorant l'activité photosynthétique, en augmentant son absorption de l'eau et en améliorant la structure des propriétés du sol, sans oublier son rôle dans l'activation du système de défense et de protection contre les dommages oxydatifs. De même, la symbiose des PGPR ont un effet positif sur l'adaptation des cultures contre les stress environnementaux, tels que le stress hydrique, avec plusieurs mécanismes tels que la production des phytohormones, la production des sidérophores, la synthèse d'ACC-déaminases, solubilisation de phosphore, production des exopolysaccharides. Pour ce qui stimule la croissance et la photosynthèse des plantes, améliore la production des métabolites secondaires ainsi que augmenter la résistance au stress hydrique. À la lumière de ces résultats, nous avons ciblé à évaluer premièrement l'effet d'application seule ou combinée des champignons mycorhiziens des PGPR sur la qualité des graines de quinoa sous stress hydrique et en même temps évaluer l'efficacité de ces biostimulants appliqués séparément ou en combinaison sur la tolérance du quinoa au stress hydrique. Quant au matériel biologique, on a utilisé les graines du quinoa, un consortium de 2 souches de bactéries PGPR et un complexe mycorhizien autochtone. Passons maintenant au protocole expérimental. 8 traitements ont été appliqués au total avec un traitement témoin, un traitement avec l'application du consortium mycorhizien, traitement avec l'inoculum PGPR et traitement avec la double combinaison de ces 2 biofertilisants. Après la germination des graines, l'application des champignons a été appliquée lors du repiquage des plantules, suivie par l'application des PGPR. L'application du stress a été réalisée après 2 mois et la récolte a été effectuée après 4 mois de culture. On a mesuré plusieurs paramètres, à savoir les paramètres de croissance tels que le poids sec des graines, le poids de 1000 graines, la teneur des sucres, teneur des protéines, les teneurs en flavonoïdes, polyphénols, ainsi que l'activité des PPH. Nous avons évalué les teneurs de marqueurs de stress tels que le peroxyde d'hydrogène et le malondéaldéhyde. Passons maintenant aux résultats. Comme vous pouvez constater, le stress hydrique a impacté négativement le poids sec des graines ainsi que le poids de 1000 graines, tandis que l'application des biostimulants a augmenté significativement ces 2 paramètres. De même, pour la teneur en protéines, le stress hydrique a diminué négativement la teneur en protéines. Pourtant, l'application de l'inoculum bactérien a augmenté ce paramètre par 47% pourcentage d'amélioration par rapport aux témoins stressés. Pourtant, le stress hydrique a boosté l'accumulation des teneurs en sucre, surtout chez les plantes non traitées. Quant au système antioxydant, les teneurs en polyphénol, en flavonoïdes et l'activité des PPH a augmenté clairement par rapport sous stress hydrique plutôt par rapport aux plantes non stressées, surtout chez les plantes traitées avec l'inoculum bactérien et celui avec le consortium mycorhizien. Pardon. De même, l'impact négatif de stress hydrique se voit clairement. Il a augmenté les teneurs en malondéaldéhyde et en H2O2. Pourtant, l'application des biostimulants seuls ou combinés a pu réduire les teneurs de ces deux marqueurs de stress. Donc, on peut constater clairement l'effet dépressif du stress hydrique sur la croissance et le rendement du quinoa. Pourtant, l'application des biostimulants a augmenté les teneurs des antioxydants dans les graines, ce qui a amélioré significativement la teneur de ces plantes sous un stress abiotique néfaste. Et merci de votre attention.
Thank you, Ms. Wissal. I think you still have a few minutes. Do you want to make like a summary in English or no? For your presentation, just like 1 minute or
Okay, after questions?
no, right now. We still have time. Go ahead. That's what
Hello. We in this presentation we evaluated the impact of two biostimulants, PGPR and EMF, mycorrhizal fungi, on growth and the quality of seed of quinoa under water stress. After our experiments, we can see clearly that the impact of these biostimulants increased significatively the antioxidant systems in grains of quinoa and boost the productivity and tolerance of plants of quinoa under this abiotic stress, such as deficit hydric or stress. Thank you.
Thank you very much. So we're gonna follow with our next presenter. So in the program you have Professor Sharqi Ghulam, but he decided to pass his presentation to Mr. Mohammed Redwan Bourahimi, who is like PhD student. The floor is yours. You have 12 minutes.
Thank you so much, Professor. Ladies and gentlemen, I am very honored to present in front of you today one of our recent research regarding a synergistic strategies to enhance the tolerance of the quinoa under high level of salinity stress under greenhouse conditions. So, as you all know, salinity has become one of the very severe inconvenience all over the world. And as you can see, Morocco is one of the examples. And as you can see, all of Morocco has, its soil has affected with salinity. And by the way, this picture here was modified in 2022. So it is expected to rise in the past 3 years. And to adapt to this inconvenience, one of the promising strategies is to use or adopt salt-tolerant crops. And ladies and gentlemen, the quinoa is an excellent example. Not only for its moderate tolerance to salinity stress, but also for its higher nutritional value. It is very rich in protein, dietary fibers, and essential minerals. and most of all, it is naturally gluten-free. Therefore, its value or its economical value, it is rising in the global market. However, despite its moderate tolerance to salinity stress, its performance and productivity still highly affected under high salinity stress levels. Like, for example, in our case here, we will under 200 millimolar of NaCl. Thus, we will try to use different combinations between three strategies: seed priming, the biostimulation, and the application of soil amendments on enhancing the quinoa's tolerance to salinity stress. So our experimental design will be starting by priming or pre-treating our seeds, our quinoa seeds, and we chose the ICBA Q5 varieties that were grown in agronomic soils mixed either with phosphogypsum or cattle manure. Regarding the priming and the biostimulation treatment, we used both the zinc sulfate and sodium metasilicate, but at different concentration for each treatment. Regarding our experimental design, so we included a positive control, which is a witness to the optimum condition with no stress, and followed by a negative control, which is, of course, stress with no treatment. And we tried to apply all the possible combination between the priming zinc sulfate and the priming with zinc sulfate or sodium metasilicate, the biostimulation as well, and both the amendment, either with double or triple combinations. So in the 20 seconds, we started by sowing 25 seeds in each pot. And when our plants or our seedlings reached the stage of four true leaves, we started applying the salinity stress progressively, and also we kept only 5 plants with the same height for each pot. After 4 weeks, we started applying the biostimulation for 4 weeks, once a week. And about 80 days from sowing in total, we took some in situ and agrophysiological measurements, and in the total of 90 days, we harvested the plant for further measurements and assays. So as you can see, like I mentioned in our objective, salinity has a very negative effect on quinoa plant under high salinity stress levels. Its dry biomass dropped by up to 77% compared to the optimum conditions for ICBA Q5 variety. And as you can see, all of our treatment has significantly improved this parameter, and especially when for the triple combination between the biostimulation with zinc sulfate and the priming with both element and for both soil amendments. Same thing for the electrolyte leakage, which is an interesting marker for cell membrane damage. And as you can see, normally it will increase the electrolyte leakage percentage. It will increase under salinity stress condition. And in our case, it was increased by up to 128%. However, all of our combination that we have tested gave a significant decrease of this parameter, especially in the combination of biostimulation with zinc sulfate, with the presence of cattle manure and the priming with zinc sulfate as well. This double combination between the cattle manure and the priming with the SE that has the lowest decrease. For the glycine betaine content, which is a very, very important osmolyte that plays a very vital role in the osmotic adjustment, which normally will increase under stressful conditions. Like in our case, it did increase by 51%, 51% actually. And for all the tested treatments, all the combination, we had a more significant increase by another 50% compared to the negative control. For the nitrate reductase activity, which is an important enzyme that plays a vital role in nitrogen metabolism, this enzyme is very sensitive to salinity stress, which you can see its activity dropped by up to 63%. And same case for all other treatments. All the combination, especially the triple combinations that are related to zinc sulfate, gave a boost to this activity by up to 90%. To summarize, in general, like you saw, ICBA variety has very affected negatively due to salinity stress, and the application of the combination between these strategies, either by double combination or triple combinations, gave us a very significant increase compared to the untreated control. And here we marked the most important increases regarding the triple combinations, since it gave the best result compared to the other double combinations. Just I want to highlight in the perspective of this study was done in greenhouse conditions. Of course, they are controlled conditions, and as perspective, this should be also will be done this should be done in the field conditions to confirm the viability of this strategy, since the greenhouse conditions are not the same as the field conditions. And thank you for your attention.
Thanks, Sir Mohammed, for this very, very interesting presentation. We'll keep questions at the end of the session. So I invite Ms. Habiba Sood for the next presentation on the effect of tillage and mulching on water productivity. The floor is yours. If you can make it in 10 minutes.
MashaAllah, 10 minutes. Thank you very much. I'd like to thank in the beginning, the organizers for giving us this opportunity to talk a little bit about our work in Kuwait. So I know this presentation just before lunch, so I have to make it very simple with little data just for digestion. So I am Dr. Habiba Al-Mnaya. I'm a senior research scientist at the Kuwait Institute for Scientific Research. Actually, my focus is mainly on development of mutant crops. I worked with the IAA for like more than 12 years, and we already have more than like 10 lines on sorghum bread, and also we have barley, more than 6 varieties now. It's ready to register. So I thank Ms. Fatma for the support from IAA for doing all this research in mutation breeding using gamma ray and other facilities. actually, I'm not only working in mutation breeding, but also we are working, I think I need to use this one, it's better. Is it, you can hear me? How to open it? Yeah, okay. So also we are working on the use of smart agriculture. So we are not only producing the seed or not producing the varieties, but also we are trying to find the best method how to grow these varieties under our climatic condition. So we are using the smart agriculture. Mainly we are using this one, the establishment of optimized soil, water, and nutrient management practices using a nuclear and isotopic technique like N15 and other isotopes. I published more than 60 publications in refereed journals and also in conferences. If somebody don't know Kuwait, Kuwait is actually Arab country. It's in Arab Peninsula, and it's has very fragile ecosystem, and the temperature is very high. During summertime, it's more than 50 degrees, maybe 55 even, and it's very harsh for a plant to grow under this condition. So if we don't have our local breed, it will be really difficult to produce any crop under this condition. And also, as we have very limited water resources, and the soil is very sandy, so it needs lots of work to produce these things. Okay, now this work, part of a big project. Big project is producing the seeds, then application of the smart agriculture. So in this presentation, the goal is to enhance water productivity. Enhancing water productivity, what does it mean? Is we need little water to produce a crop. This is the major thing. So if we don't know the method how to reduce the water, it means we need drought tolerance crops, or maybe different practices, agriculture practices, to have good production with little water. So the water productivity is a key metric in crop production in arid region with limited water availability. So if we have high water productivity, so it means more crop per drop and more efficient water use. Our target is to maximize yield without increasing water use. So it's efficient use of our limited resources, because we have very limited water resources in the country. So it means if we are applying this water productivity, the benefit is efficient use of our water resources, in addition to higher yield without increasing the water use, and support sustainable agriculture in the country. So I have almost four applications or 4 practices used for this production under our breed. The main one, which is we try to use conservational agriculture with our crop. So we have conventional, which is the farmer always using. We are trying to move from conventional agriculture to conservational agriculture with our breed. So the first thing is the tillaging, which is the preparation of the land. So we try to use minimum tillage versus the normal tillaging, which is the farmers always using. Because tillaging improves water infiltration and the storage, helps root access deeper moisture, improves soil structure and nutrient availability, and enhance water productivity, effective when combined also with mulching. What is mulching? Also, we are using mulching for our crop as one of conservational agriculture practices with our crop. So mulching, it reduces evaporation from soil that's why I said we have very high temperature in Kuwait. So if we are using mulching, this will improve the soil, will regulate the soil temperature, improve soil moisture, lead to higher water use efficiency, improve soil structure and fertility, reduce soil erosion, and it's actually lots of benefits of using the mulching. In addition to that, the fertilizer is very important to reduce the fertilizer use under this harsh condition. So we try to use the nitrogen less than the nitrogen which the farmers always using. Also, the irrigation rate is also affecting the crop performance and water use. So now, if we go to the main experiment. Actually, we use barley. Also, I'm working, as I said, on sorghum, but we use barley in this study because it is a model species. Easy to study the barley genome, easy to study the barley life cycle is very short, so it's easy to work with the barley. That's why we use barley to increase the water productivity. So the aim is the ultimate goal of this study was to enhance barley productivity while conserving vital water resources in arid region. So all the things is we are trying to reduce the water use. This is the main thing because we don't have water. This is the major issue. So the objective is to determine the interactive effect of tillage, mulch, irrigation, nitrogen application rates on barley water productivity. Then to identify the best combination of all these practices. So this is actually, if you can see, conventional and conservational. We use tillage and non-tillage. So if we can check here, this is conventional tillage with mulch, conventional tillage without mulch, conservational with mulch, conservational without mulch, and then we use a number, it's 3 applications of irrigation and 3 fertilizer application. This is, as I said, is part of a bigger project. During this project, also we use isotope, which is N15, to determine what is the nitrogen use efficiency for this experiment. But because of the timing, we just use this one. So you can see here, this area is without any tillaging, and this one with tillage, the normal tillage with the farmer use. Then this is all the experiment, how it is done. So here you can see this one with mulching, and that one without mulching, and this one with mulching. And here you can see the tillaging and how is the soil really is changed. The structure of the soil is different than the structure of the soil which we actually without this application. So here you can see both irrigation, nitrogen, both the interactive effect of water productivity of barley. So highest productivity recorded under 50. If you can see here is 4.6 and 4.17. It means with 50 irrigation and 50 nitrogen, it will be better production if we are using the mulching. And this is without mulch. Also, actually, it's that the soil is improved because with using practices, agriculture practices, it reducing the amount of water which is evaporated. So this one, actually, this conventional tillage without mulch. I want to go to this reduced tillage with mulch. You can see when we use reduced tillage with no tillaging and with mulching, the production was increased because the water is reduced and also laborers were reduced. So this is the better. Here is water productivity was significant influence by both irrigation and nitrogen. This is reduced tillage without mulching. this is the different graphs. I don't think we have time to go through all this one. So the result of this one, deficient irrigation recorded higher water productivity for all the treatments. And then the highest water productivity was recorded under conventional tillage with mulching and under 50 irrigation and 100 nitrogen per hectare. The water productivity was increased by 100% in comparison to control. This, if we use conventional tillage and non-mulching. Barley recorded 79% increase in water productivity with conventional tillage with mulching, under 50% irrigation and 50 nitrogen, comparison to its control. So the treatment with highest water productivity, conventional tillage with mulching, 50% irrigation and 100 nitrogen increased than the control. Conventional tillage with mulching and 50 irrigation, deficient irrigation and 50 nitrogen reduced 79. So selection depends on the cost of water, nitrogen, and market price. Yes, so in conclusion, improving water productivity is an important policy in arid region. So that's why we need to use, even if we are producing high productivity seed, we need to use good agriculture practices. Smart agriculture is very important to use with any seed produced under harsh environmental condition. So that's why under harsh environmental condition, we need to use mulching and we need to reduce nitrogen, using nitrogen use efficiency to reduce the use of the water. Thank you.
Thank you very much. Thank you very much, Dr. Habiba, for this presentation. I was told that we don't have time for questions, so I invite you to talk to the presenters, and we'll have another session this afternoon. If you guys here, we can start with like 5 minutes. We can borrow some time from the afternoon session, and we can ask questions to the speakers. But now we want to go for lunch. Thank you very much.